emitting light that you cannot, they're turning light you cannot see into light that you can see. Right, but we're not talking about fluorescence, we are talking about dyes. Okay, benzene. The benzene ring has resonance. That means it flicks between this structure and this structure, centric structures which can form. But basically, these electrons are delocalized around this ring, they move around. The more free electrons there are, the better something, the more light something will absorb, generally speaking. So these delocalized electrons are really good for absorbing electromagnetic waves, and that's what makes the compound colorless. So aromatic compounds are simply compounds that contain a benzene ring. Benzene rings have resonance. Resonance is important for absorbing light, which allows us to perceive these compounds of different colors. For certain molecular arrangements can affect the light that is absorbed and the light that's reflected by these compounds, and that produces color. A very frequent arrangement of this type is the quinonoid ring. A lot of dyes contain this. Paraquinonoid rings, any compound that contains this is colored, and we'll go through some examples in a moment. So this is a paraquinonoid ring. The simplest parabenzoquinone is the simplest arrangement of this substance. So it's simply the benzene ring, but has these two double bonds leading out with these oxygens attached to them. These are carbonyl bonds. So these are double bonds here. And what you can actually get in this arrangement is delocalization of these electrons all the way through this ring and all the way to the oxygen. Okay, so these double bonds can flip around, which means that it absorbs light particularly well, reflects light particularly well, and you get color out of this. So this is yellow solid. You make a pale yellow solution when you dissolve it in water. But this is not a dye. It's a colored solution, but it's not a dye, because it doesn't have any affinity to any tissue constituents. So if we were to just stick this onto a tissue slice, a tissue section, it wouldn't colour it at all. It would just wash straight off. It has no affinity to any tissue constituents because it is not charged. There's no overall charge on this. There's nothing for it to interact with the tissue with. So how do we go about changing that? So what we do is we put an auxochrome on the dye. An auxochrome, which is described in a bit more detail on the next slide, please, is something which can interact with tissue. So we call an auxochrome is effectively a colour helper. So, for example, ferrarosaniline is, if you link all of these moieties together, double bonds in this down here, but don't worry about that, you get this structure here. This structure here is coloured. Okay, this is the quinonoid ring here. It's coloured. And critically, there is this positively charged amino group down here. And this is a coloured compound that can link with tissue components. It has resonance because these double bonds can flick around. Okay? Resonance between this structure and that structure. That means it's good at absorbing electrons of a particular wavelength, absorbing photons of a particular wavelength, which means that it will have a color. And the other essential component is this NH2. NH2 plus, it's amino group here, well, it's not really an amino group, it's got a double bond there, but this NH2 plus group here, because this is now charged, it can interact with negatively charged tissue components. So this will be a dye. Okay. The two words you need to know here are chromophore. Chromophore is the chemical moiety that has the colour. And the auxochrome is the chemical moiety which will allow it to attach to a tissue. So a dye must have both of these. It must have chromophore and it must have an auxochrome. So this is that the chlorosanilene here, shown with the chromophore, and then we'll bring here, and the auxochrome shown. Remember that these other structures have an influence upon the colour. Okay, so they are also effectively part of the chromophore because the resonance of the electrons goes all the way through this entire compound. So without these two moieties up here, this will not have the same colour. So whilst the colour is determined by the fact it's got the benzene ring in, the other things that are attached to it determine which colour that is. I'll give you some examples of that in a moment. Okay, so auxochromes, there aren't very many that are used. One of the most common ones is the NH2+. This is a basic cationic auxochrome. And as I said earlier, dyes generally salts. So for a salt, you have a positively charged dye, which means you need a negatively charged ion to go with it to make it a salt. So just as sodium chloride is positive sodium and negative chlorine. So this dye with this auxochrome will be positively charged dye and a negatively charged ion to go with it. For example, chloride. You can also have sulfate. You can also have acetate. You can get anionic or acidic dyes. So these are negatively charged. So the negatively charged ion is coloured. You need a complementary positively charged cation to make the salt. So that would be sodium or potassium, generally speaking. So the negative charge auxochromes that we get, sulfonic bonds, hydroxyls, carboxyls. These ionize to give these negatively charged species which will interact with positively charged tissue components. Okay. So I mentioned that while the benzene rings are vital for the colour to exist, the actual colour that you get is determined by which modifying groups you have on the dye. And so, for example, fuchsin is a mixture of these two compounds. It has its colour because the rosanilene and the chlorosanilene differ by this methyl group. And this methyl group changes the absorbent properties of the dye and changes its colour. So this methyl group makes this dye more blue. It changes the wavelength of the light that it absorbs and reflects. Another example is crystal violet. Crystal violet is used to be used as a DNA stain a lot. I'm not sure how commonly it is used these days. I haven't used it for years. Crystal violet, chlorosanilene, which has methyl groups, two methyl groups attached to each of these nitrogens. Still positively charged, so it can it's still a dye. It still has this auxochrome here. The chromophore has been modified by the addition of these methyl groups, and that makes it a purple dye. Okay. So far, we talked about resonance. We talked about the fact that dyes are charged. We talked about chromophores, auxochromes, and modifiers. Right. That is some of the basic chemistry of dyes dealt with. Anybody got any questions on that bit? Okay. Everyone's still feeling enthusiastic. You look enthusiastic. Yeah, I can tell. Right. Great. So, we are now going to talk very briefly about a few quinonoid dyes. Before that, we are going to talk a little bit about carbohydrates. So in the context of cellular pathology, we're interested in two main carbohydrates, glycogen and mucins. We'll talk about glycogen first, mucins we'll talk about later on in the lecture. So glycogens, lots of glucose molecules linked together. the major rapidly mobilizable carbohydrate source in animals is glycogen. Okay? You have large deposits of it in your muscles and in your liver. There are various different forms of glycogen that you can find in the liver, and critically, the distribution of glycogen can change in certain pathological conditions. So, for example, various different carcinomas, which are listed on here. I don't need to read out for you. Okay, so how do we look for glycogen? We look for glycogen by modifying it. So the glycogen, I believe, is digested by diastase, which is an enzyme, and then the enzyme cleaves glycogen into simple carbohydrates. Then use what's called periodic acid. And that breaks the glycogen at this particular point, breaks the carbon-carbon bond. And this exposes these two carbonyls, which can then react with something called the Schiff's fusion sulfurous reagent. Okay? And this forms this structure here. And you see we've gone from a situation where we only have single bonds here. Single bonds only have a pair of electrons linking these two. To two double bonds down here. Okay? So there are, see over here, two double bonds. There are considerably more free delocalized electrons in this system, which then makes this a colored compound. It allows you to see it. Okay, so this is the way we can change something which has no color into something which has a color by introducing these double bonds into it, which can then absorb the wavelengths of light. And this results in a magenta product. So we can visualize glycogen, for example, basal lamina. It's important to identify the basal lamina when you are looking at tissue sections, especially if you're looking at tumors. Okay. This is an example of an acidic stain for mycobacteria, the Seal-Nielsen stain. Mycobacteria are parasitic bacteria that live within your cells. An example of that is leprosy bacteria or tuberculosis bacteria. Obviously, you want to identify them within living cells. This is what we call an acid wash stain, the Seal-Nielsen stain. It's what we call a special stain because it's designed for a particular purpose, one specific purpose. We'll talk a bit about those towards the end. So this is useful in identifying things such as leprosy. Okay. Right. Now we're going to come on to probably the two most important dyes, stroke stains in histopathology. So the first one we're going to talk about is hematine. Hematine is actually derived from this tree. Okay? So it's a natural compound, it's been known for a long time. It's a brownish red colour and becomes blue at high pH. Great for a pH indicator, that's no use as a dye at all. It requires, in order to become a dye, because it itself cannot link to tissues, but in order to become a dye, it requires a mordant. Okay, so a mordant is effectively a dye-helping reagent. It is something that links a dye to a tissue, and by so doing, bestows colour upon the dye. It's like a sort of adapter molecule. And when you do this with hematine, it becomes hematoxylin, which is one of the most important dyes used in histology. So it comes from the heartwood of this particular tree, which is why it's called hematoxylin. So what is a mordant? So basically, a mordant is something which binds to the tissue, particular tissue feature, and also binds to the dye. So that forms this bridging reaction between the tissue and the dye. And when that happens, you get what's called a dye lake being formed. So a dye lake is a repository of dye bound to the tissue by a mordant. This is something where we don't fully understand the chemistry, but it's probably something to do with complexing ions. All mordants that we use commonly, contain metal ions. And metal ions can form complexes with a variety of different ligands. Some examples of them are potassium alum, ammonium alum, iron alum, chrome alum. And if you're really interested, these are the chemical formulae of those particular mordants. You can see each of them contains a metal ion. And metal ions carry a positive charge. Okay, so how does this work with hematin? So this is hematin. Okay, no overall charge, not going to bind to anything. However, it contains this hydroxyl group here and this carbonyl group here. And the important thing to remember about oxygens is every oxygen atom has on it what we call a lone pair of electrons. So a pair of electrons which are unpaired. So there's a small patch of negative charge on every oxygen atom. Okay? So what happens when we put our mordant in? So one of the theories behind this is that when we put our mordant into this, so take the example of the aluminium alum here, okay? This forms a complex ion by which these lone pairs of electrons are attracted to the positively charged aluminium ion here, okay? The rest of the spaces on this complex are taken up by water, and this then interacts somehow with acidic components of the tissue, because the aluminium ion again is positive. So if you like, you have an acidic component of the tissue which is negatively charged, for example, DNA. That attracts the aluminium to it, and the aluminium attracts the hematin. So this aluminium is forming a bridge between a negatively charged tissue and the hematin, and this forms hematoxylin. It's possible that eventually you get covalent bonds being formed between the aluminium and the tissue, because this dye persists for a very long time. It's very, very stable. And what it does is, probably some slightly better versions of this you can see later, but you can see the nuclei of these cells here are sort of a bluish purple colour. Okay? So hematoxylin is used to stain, in fact you can see it much better down here. Hematoxylin is used to stain the nuclei of cells. Okay? Because it stains negatively charged tissue components. And the nucleus contains DNA, and DNA, as we all know, is based on a phosphate backbone. The phosphate is negatively charged. Okay? So hematoxylin is used to stain negatively charged tissue components, mostly the nuclei of cells. Usually, here's another example of that. So you can see the nuclei of the cells in this nice bluish purple kind of colour. Okay. Hematoxylin is usually used in combination with another dye, which is called eosin. Okay? Eosin is a xanthine dye. So it's not, it's a different sort of chemical to hematoxylin. And this is what a xanthine dye looks like. This is a skeleton formula. Right, okay. Eosin, named after the colour of the sunset. Okay, so, or rather the colour of the dawn sky, specifically after the sunset. Okay, so eos is the Greek word for dawn, and this is a red sky in the morning. Okay, so it's pink-orange colour, this dye. And this is anionic. So it is a negatively charged dye, which means it will interact with positively charged tissue components. Positively charged tissue components can be found in the cytoplasm, lots and lots of proteins in the cytoplasm. Many of them, most of them, have some basic residues. So the basic amino acids are arginine, lysine, and under some situations, histidine. Okay. And so eosin will stain these components. These two are very often used in combination because one will show you the nuclei of cells, one will show you the cytoplasm of cells, and the combination of the two will basically allow you to see the cellular structure of the tissue that you're looking at. And this is one of the most basic dyes used in histology. It's very useful for looking at morphological features. Some more examples. These look like muscle fibers to me. Polynucleate cells with all the protein filaments stained in pink by the eosin. So this is the structure you would expect to see in this tissue, but if in your biopsy you see something different, you know that there is something going wrong. And that's quite often the way that we use. Okay. So the basics of the interaction of dyes and tissues. It's the very simple electrostatic chemistry of opposites attract. Okay? So the acidic tissue constituents are negatively charged. DNA, chromatin, RNA, cartilage, some mucins, especially weakly acidic mucins, and some lipids are negatively charged. Some lipids have no overall charge, some are negatively charged. The basic tissue constituents that you find: collagen, so these are positively charged tissue constituents, collagen, red blood cells, granules of eosinophils. So you can see this word eosin appearing in eosinophils, okay, because they're stained very nicely with eosin. There are some tissue components which we call amphoteric, so they're neither acidic or basic. So the cytoplasm of the muscle, they don't have an overall charge. However, within them, there are charged species of both acidic and basic species. Depending, this means that the overall charge of this particular area, of this particular component, will be determined by the pH in which you put it. Okay? So something which has no overall charge, if you put it in an acidic environment, will become negatively charged. If you put it in a basic environment, it will become positively charged. Okay, so the acidity of DNA and phospholipids is due to the ionization of phosphoric groups. We know this. I mentioned this. The phosphate backbone of DNA is negatively charged. The acidity of mucins is due to carboxyl groups, maybe. Some of them have sulfate groups on them. And the basic nature of collagen and red blood cells is due to the basic amino acids lysine, arginine, and to some extent histidine. Okay. So, amphoteric proteins are their characters of balance between acidic amino acids and basic amino acids. All proteins have what we call an isoelectric point. That's the pH at which they have no overall charge. Okay, if you put them above the isoelectric point, then they become negatively charged. If you put them below the isoelectric point, they become positively charged. Which I believe I said the other way around in the previous slide. I'm very sorry about that. So low pH is high proton concentration. So when pH is low and things are acidic, okay, then things like lysine will become positively charged. Okay. when the pH is high, number of protons around is low, things like glutamic acid, negatively charged. So you can determine the overall charge of proteins by varying the pH. That may well be sufficient to determine whether or not they get stained by particular dyes. Okay, so two terms you hear, basophilic and acidophilic. So basophilic likes bases or positive charge. Okay? So basically anything that's acidic and anything that is negatively charged will be, by definition, basophilic because it will want to interact with positively charged things. Opposites attract. Acidophilic, being the opposite, likes acid or negative charge. So some tissue components we call basophilic. Basophilic means they want to interact with a positive charge. So these tissue components must be negatively charged. Similarly, some tissue components we call acidophilic. They want to interact with a negative charge. Okay? So these tissue components must be positively charged. Basic dyes react with acidic tissue components, for example, hematoxylin, positively charged because it's got that aluminium orbital or any other metal ion, and it reacts with negatively charged tissue components. Acidic dyes react with basic tissue components, for example, eosin is negatively charged. It reacts with positively charged tissue components such as positively charged amino acids on proteins in the cytoplasm. This is possibly the most important take-home message in this particular section. The chemistry behind it is relatively simple, so hopefully that's nice and clear. Okay, however, there's always complications to these things. So, acidic tissue proteins are not necessarily resistant to coloration by acidic dyes, and vice versa. So all proteins contain both positively charged and negatively charged residues. Some proteins are overall positively charged, some are overall predominantly acidic. Some proteins which are overall basic, so they have an overall positive charge, will still contain some acidic residues, and vice versa. Proteins which have an overall negative charge will still contain some basic residues. Okay? So all proteins in a cell will eventually be dyed to some extent by a dye, regardless of whether it's acidic or basic. So we have to control this staining, this dye reaction. Okay? We do that by the amount of time that the stain is put on for. That's one way of doing it. So, a cationic dye will preferentially stain negatively charged tissue components. So if we only put it on for a short period of time, it will only stain the negatively charged tissue components. Vice versa, an anionic dye, a negatively charged dye, will stain the positively charged components of the cell first. Okay? So if we control the amount of time these dyes go on for, we can control the specificity of the reaction. We can control, as I suggested, amphoteric objects by controlling the pH, and that changes the charge of the objects. Remember, these cellular components are positively charged or negatively charged depending on the pH of their environment. And so you can arrest the dyeing before it starts to stain components that you don't wish to stain. That's called progressive staining. There's also regressive staining, whereby you stain everything and then you wash out the staining on the components you don't want to stain. And obviously the things with the lower affinity of the dye will de-stain first. So progressive, you control the amount of time you put the dye on for. Regressive, you stain it all and then you wash out everything you don't want to stain. Okay. So as well as ionic interactions, which is what we've been talking about, acid-base interactions, what we've been talking about up until now, there are three other types of forces by which dyes can interact with tissues. I'm sure you've covered these all probably in the first year of CBGs. So covalent bonding, which there are likens to getting married, wedding ring. Okay, so covalent bonding is a direct bond between the dye and the tissue component, and it's thought that eventually this is what happens in toxin staining, covalent bond between the mordant and the tissue component. Hydrogen bonding is bonding caused by an interaction between the electronegative oxygen and the electropositive hydrogen, which are found in OH groups or in water, or in NH groups, or in a wide variety of cellular components. Hydrogen bonding is probably the most important force in all biology. It's the force that allows water to be a liquid at room temperature. And then there are also van der Waals forces, and these are the forces by which things with no overall charge, no overall electrostatic difference interact. Things such as oils and fats. The reason why oil and water don't mix is water can form hydrogen bonds and oil can only form van der Waals interactions, which means the two of them can't interact with each other. And various dyes work in these ways. We'll go through a couple of examples later. So hydrogen bonding, for example, there are two different hydrogen bonding stains here. This is a carmine staining for glycogen, and this is a Congo red stain here for amyloid. Amyloid is a protein aggregate found generally in the brains of people with Alzheimer's disease. Okay. Right. We're doing quite well for time. I think we're going to have a 10-minute break now. Everyone happy with a 10-minute break? Or does everybody want to press on? No 10-minute break. Okay. Great. We'll do that. And then we'll be talking about azo dyes after a little break. Okay, so you'll be back here at 5 to 10. That would be brilliant. Anybody got any questions? I'll be hanging around here.
Even in like, do you know, like the traffic light just doesn't move. Yeah. Well, we've already done one.
What do you mean? It's like staining them.
It's all doing a practice, basically.
These are what I'm saying. There are a couple of slides here. Dyes are not apart from that. Dyes the same, different. Dyes. It's lectures on Blackboard. It's been on Blackboard for about the last 5 days. So, especially after the conversation. Because I'm not really involved in
So you've got time most.
I can manage. Why did have someone? Why you?
Did you not like get tea in the trial? No.
I basically the second I just walked in that is like you live.
You're already downstairs.
Yeah, that's
Like he had flashbacks to some kind of war. The great PG war. Scolding hot fan. Sort of teabagging the scarlet for life. But the same, because I used to drink tea like, I used to make tea in bottles when I was a baby. Loved it. I got all the time, not like every time.
I got to keep it up.
But that's a lot of it.
Short fuck, and I'm going to hold your kid home. And she wants to try it. Try it. And yeah, she's like,
I used to be like that one kid at the truck shop at after school club. When you're like, hey, look, I'll have a tea, please. I'll have a cup. Definitely found milk. Can't smoke my pipe with them. I've never tried that actually. Might be really nice.
We go like
remix and just like a cover of it.
like a serial that goes well.
I might try it.
Once tried.
Orange.
I'm like easy one. So the speaking orange. I will talk about it. Look at that. I always wanted to make like frozen tea with it. Tea. Just stick it in.
Yeah.
Anybody seen the register around somewhere? Who's got that? There's something right at the back here. No. The whole class is going to get zero attention. Right, okay. So, different. Has anybody in here not signed the register? Okay. Did it get round to this side? Obviously, you haven't got round to this side yet. Okay. Start there. We can just pass it down to the front when you finish signing it, and I'll take it out to these guys over here.
5692. Strangers. That's not my student. I know all of the important ID numbers that identify like my national insurance.
I know. Scream it out.
Right, okay, I think we'll start again so we can get finished in time for you to have a decent break before the last part of the session at 11 o'clock. Okay, so remember at 11, David Quadro is going to be here and give you some information about coursework, and I think various other bits and bobs about the module. So it's really important that you're here at 11, okay? Then he's going to do the third part of this lecture, which is about immunocytochemistry, I believe. Right, so we've talked about why things are coloured, we've talked about how they interact with tissues, and I give you some examples of some of the most important stains and dyes used in histology. Okay, so there are three main ways of staining tissues, as I said. There's dyeing, there's a solution of the lysochrome. Some people might call this a stain rather than a dye. And then there's local formation of a coloured substance. We're going to talk about a solution of a lysochrome. So lysochrome. Lyso means solution, chrome means colour, so it's a coloured solution. And the majority of these are azo dyes, and they're often used to stain fats. There's one particular example I'm going to give you in a moment. Those used to stain fats. So examples of azo dyes. Often these are anionic dyes, such as orange G or Deerbush Scarlet. The azo refers to this nitrogen-nitrogen double bond. See? Nitrogen-nitrogen double bond, which links two aromatic rings together. Okay? And this can occur several times in the dye. This results in delocalized electrons and resonance that allows absorption of photons of certain wavelengths and reflection of photons of certain wavelengths, causing the dye to be coloured. Okay? So that is the same principles that we were talking about earlier. If they have one azo bond, they are called monoazo, 2 they're called diazo, 3 they're called triazo. Nice and simple. So not all the azo substances are dyes. Okay? So an example is Sudan 4. It has the azo chromophore and it's coloured red. So it's nice red colour. However, it doesn't have an auxochrome. So remember, a dye has to have a chromophore and an auxochrome. So although Sudan 4 is coloured red, it doesn't interact with specific tissue components. It can't bind to them because it doesn't have the auxochrome. It's insoluble in water. What does that tell you? It tells you it's hydrophobic. It cannot participate in hydrogen bonding. Okay? But it's very soluble in fats. So that actually makes it quite useful for staining fats. So what you do with Sudan 4 is you dissolve it in a solution of 70% ethanol. Ethanol is a very interesting solvent. It has an OH group on it which can interact with water, which is why ethanol is soluble in water. But ethanol is also soluble in fats because it has another end to it which is non-polar, which can interact in van der Waals interactions with fatty compounds. So you can dissolve Sudan 4 in 70% ethanol. And what you do, then you wash that nice red solution, which is dissolved in ethanol, onto your tissue. And because the majority of your tissue components are aqueous, remember we are 70% water, the Sudan 4 cannot dissolve in water, cannot interact with it. So it doesn't go into other tissue components, it specifically goes into fats. There we go. So it goes from the 70% ethanol solution into the fats. It interacts better with the fats than it interacts with the ethanol, which means it's preferentially taken up out of the solvent and into the fat tissue that we're wanting to stain. Example of that. So Sudan 4. This is what Sudan 4 looks like. As you can see, no charge anywhere on it, which means it cannot interact with water. It's insoluble in water. But it's got all these aromatic rings, and it's got 2 azo bridges, which gives it a nice colour. It has no overall charge. It has a lot of capacity for van der Waals interactions to interact with fats, and it will stain the fats in this red colour that you can see here. It looks a bit better on my screen than it looks up there, unfortunately. Okay. As well as azo dyes, we have thiazine dyes. So these are generally cationic, so positively charged. And they have 2 aromatic rings, and in this case they're linked by sulfur and nitrogen. And these dyes have a peculiar property called metochromatism. So they're metochromatic dyes, which means that they can impart different colours to different substances. Examples of these are methylene blue and methylene green. So what is metochromasia? So dye ions are usually dyes are taken up without loss or change of colour. That's something important we talked about earlier on in the lecture. A dye has to be able to interact with a tissue component without its colour being lost or being changed. But dyes which exhibit metochromasia, they change their colour when they interact with particular tissue constituents. So the dyes, unmixed dyes, have a different colour to the dyes which, after they've interacted with particular tissue constituents. Okay. The dye is then called metochromatic. And the tissue constituents that we dye with these are sometimes called chromotropes, which means colour terms. Chromotropes, not to be confused with chromophores. So, chromophores are things which produce colour bearers, chromophores. Auxochromes are things which are colour helpers, and chromatropes are things which change colour when they bind to particular constituents. So all the metachromatic dyes we use in histology are basic. And the colour shift caused by the chromatropes is always in the same direction. So the wavelength becomes shorter. Okay? That means that red dyes will become yellow, green dyes will become bluer, blue dyes will become purple. And this is called the hypsochromatic effect, hypsochroma effect. Shift towards shorter wavelengths. So here is the visible spectrum, again expanded. So longer wavelengths are red, shorter wavelengths are purple and violet. So basically, if you have a metachromatic dye, say it's red, but when it binds to tissue components, it gets shifted to produce a yellow colour. Similarly, a green dye, we shift to produce blue colour, and a blue dye, we shift to produce a purple colour when they bind specific tissue constituents. And this is quite useful for identifying certain things. For example, histamine in mast cells. So mast cells, an important part of the immune system. Histamine is an important inflammatory mediator. And histamine is contained in granules. Okay, so secretory granules, tightly packed in secretory granules. These histamine cells also contain, obviously, they have nuclei. And because this is a cationic dye, it can bind to nucleic acids. So these nucleic acids have the orthochromatic color blue. So this is a blue dye, toluene blue. It stains the nuclei of the cells in blue. And it stains sulfated polysaccharides purple. This is because the groups are closely packed together, and when the dye comes very close together, you get the metachromatic shift. So this is an example of how metachromasia can be used with one dye to stain two different components of a cell, different colours. The dye binds in the monomer to nucleic acids, blue, or it binds in the polymer, so closely packed together negative components, it turns purple. Okay. Right. So we're going to talk about tissue permeability now as a way of differentially staining. So the amount of dye taken up by an object is not just about what it can interact with, it's about how easily that dye can get into the object in question. So how easily permeable the dye is into that particular tissue. Okay, so you can have differential staining of different tissues due to the differences in permeability. And this is usually, if a dye is a very large ion, it can get easily into less densely packed tissues, but not very easily into closely packed tissues. So it allows us to differentially dye objects that carry the same electric charge. So, for example, both collagen and red blood cells carry a high percentage of basic protein. So they're acidophilic. A cationic dye will bind to both of them. Okay? So the colour wouldn't be different just using one dye. However, if we use two different acidic dyes, sorry, when I said cationic, I meant anionic. So they carry basic proteins, and so they'll be dyed by acidophilic, so they'll be dyed by anionic dyes that have a negative charge. Okay? So we have two different negatively charged dyes, but of different sizes, of different ionic sizes. For example, methyl blue and hyoscine. We can differentially stain them depending on their relative permeability. So methyl blue is a very large ion with aggregates. Hyoscine, very small ions. Collagen is a loosely textured component, not very tightly packed. So a large ion can permeate it quite easily. So it gets stained the methyl blue. However, the red blood cells are tightly textured. The methyl blue can't get in, and the hyoscine can, so it stains them a different colour. So this means that collagen is dyed blue, red blood cells are dyed orange or red. This is an example of different tissue permeabilities of different components. So collagen, highly permeable. Cytoplasm, less so. Muscle, less so. Red blood cells, not very permeable. Okay? And this will be inversely proportional to the size of the ion that can get into these tissues. Okay? So large ions will not get into red blood cells, but they will get into collagen. Here's an example of how that works on a little diagram, little animation. So, the very small molecules get into the red blood cells with these three different colours. Okay. The very large, so the slightly larger dye molecules can get into muscle cells, and the larger ones, the really big dyes, so like the blue dyes, can get into the collagen part of the tissue. An example of that, trichrome staining, and that's due to the relative permeability of different tissues and cells. So, for example, red here, we've got something like blood vessels, and then the different tissue components, depending on how permeable they are, you have different colours going on. So another way of, actually, sorry, I can describe that one a little bit better detail. Okay, right. So the particular dyes on this one that are being used are a dye called Martius, MSD. So MSD is the name of the trichrome staining mixture. So Martius, scarlet blue, and nuclei blue. So it means the red blood cells here, or my mistake earlier, the red blood cells are stained yellow, nuclei are stained blue, and fibrin is stained red. The red blood cells are yellow, yeah, confusing. That's because the dye that's being used in them, not because of their inherent red colour. And the red blood cells, if you actually look at them, take them out and look at them in a test tube, they are not that really bright scarlet colour. They are sort of a dull reddish because they will deoxygenate as they take out the blood vessels. Remember that these tissues have gone through a huge amount of fixation and preparation procedures that often strips the inherent colour away from some of these things. So that's why we put these stains in to identify different components. Okay. If you want a better description of that picture, I suggest you email Sarah about it, because that's a bit outside my specialism. Right, okay, so now we're going to talk about local formation of a coloured substance. Right, so as well as putting a dye into a tissue, let's say a specific tissue component, we can also put something into the tissue that binds to a specific component and goes a colour. Now, there are different ways of doing that. One of them is using enzymes. So different tissues can have different enzymes being expressed in them. An example of this is different types of muscle fibers, which we'll look at later. So you have two types of muscle fibers. You have fast twitch and slow twitch. Okay? Fast twitch fibers are mostly anaerobic. Slow twitch fibers are mostly aerobic. So the slow twitch fibers express more of the aerobic metabolism enzymes than the fast twitch fibers. And you can use that. You can use a substance which will interact with these enzymes and create a colour substance based upon that. You can also do that by looking at things that degrade neurotransmitters such as acetylcholinesterase. And this will allow you to see different components based on the local formation of a coloured substance. Something that's quite frequently used are assays for iron, using some iron deposits. So we treat the tissue with acid ferrocyanide. ferrocyanide and dilute hydrochloric acid. Now, if there is any iron around, any ferric ions, they react with the potassium ferrocyanide and they produce an insoluble blue compound, ferric ferrocyanide, okay, which is also known as Prussian blue. Two prime examples of this. So Prussian blue is a common histopathology stain, and it's used to detect iron in biopsy specimens, okay, such as bone marrow samples. So this is a tissue stained with Prussian blue to identify chemochromatosis. And all of these blue dots here that you can see are ferric ions which have been stained blue. Blue colour has been formed at these points by a chemical reaction with the Prussian blue. So this is a classic stain for iron in biopsy tissues. You could also use this to look at iron deficient anemia. So hemochromatosis, you expect to have more iron present. Iron deficient anemia, you expect to have less. This is really important. These are asbestos bodies that you can find in the lungs of people who work with asbestos. Okay, so asbestos is the major, in fact, almost the only cause of a particularly nasty form of lung cancer called mesothelioma. And the asbestos bodies, these little fibres, when they're taken up, when you breathe them in, when they end up in your lungs, they get surrounded by iron-containing proteins. And these are known as asbestos bodies. So when we use the Prussian blue reaction here, because these asbestos bodies are surrounded by iron-containing proteins in the lungs, you get this local formation of this dark blue substance. Okay, so I mentioned using enzymes to locally produce a coloured substance. So diagnostic applications of enzyme histochemistry. Don't usually use this in post-mortem material because enzymes are not exactly living things, but enzymes are very unstable, generally speaking. So after you've done all your tissue preparations, the enzyme activity will usually be lost. Okay, so all your cells in the body, all your tissues contain a different complement of enzymes. However, if you don't treat the tissue properly when it's taken out of the person, and certainly if it's post-mortem, that enzymatic activity will be lost. And therefore, you usually need to use either frozen sections or sections which have been very rapidly removed, biopsies which have been very rapidly removed from people, and got to allow it down quickly. It's the most common use, surgical pathology. Skeletal muscle biopsies to look at, as I said, fast and slow twitch muscle, type 1 and type 2 muscle fibers, or detection of ganglia and nerves in suspected Hirschsprung's disease. Hirschsprung's disease is a disease of intestinal motility. So, there we go. This is using a chemical reaction which detects the presence of active ATPase. So ATPase is, we're talking about mitochondrial ATPase here. It is the driving force of ATP production in oxidative phosphorylation, a major ATP producing reaction in aerobic cells. Okay. And this produces a dark color. The type 1 fibers, which are highly aerobic, this is endurance muscle, slow twitch muscle, is dark here, because it has a large amount of ATPase present in it. The type 2 fibers, the slow twitch muscle fibers, so fast twitch muscle fibers, and anaerobic, the ones which are used in weightlifting, for example, or rapid force generation, are paler. The stereotypical difference in type 1 and type 2 fibers is that athletes who are long distance runners have a predominance of type 1 fibers, endurance muscle. Athletes who are sprinters have a predominance of type 2 fibers because sprinting is anaerobic. In fact, there's no need to breathe at all while running 100 meters because it's an entirely anaerobic race. This can be used to assay muscle degenerative disorders. You could also use succinate dehydrogenase. This is another part of the mitochondrial electron transport chain. And again, you can see type 1 fibers, which are the slow twitch fibers, the small diameter fibers here, are being stained with this substrate that produces a colour when it reacts with succinate dehydrogenase. The anaerobic fibers here are not producing much colour at all because they have very low amounts of succinate dehydrogenase. They tend to be larger because they produce more force. Then you have the intermediate fibers, which are a sort of combination of the two. This is a very useful way of looking. It's a very useful way of looking at the difference in the distribution of fast and slow twitch fibers in muscle biopsy. It's also, incidentally, the reason why you get different colours of meat. So red meat is predominantly slow twitch fibers, and white meat is predominantly fast twitch fibers. So red meat contains high levels of myoglobin and makes it red. It's not red because it's got blood in it. It's red because it's got high levels of myoglobin, because it's aerobic. So it requires an oxygen storage capacity. Whereas anaerobic muscle, for example chicken, doesn't require nearly as much myoglobin, so it's much paler. Here's just a little diagram of how you can differentiate type 1 and type 2 muscle fibers by assaying using chromogenic substrates of these two different enzymes and the ATPase at different pHs. That one should be on your handout, I believe. Remember, all of these slides are also on Blackboard. Hirschsprung's disease, something else we can look at, diagnose using local generation of coloured substance. So in the normal colon and rectum, there are ganglion cells, the submucosa, and these are responsible for colonic motility. So these are part of the intellectual nervous system. In Hirschsprung's disease, part of the rectum and the colon is devoid of these ganglion cells, and so you get a problem with intestinal motility. And a very commonly used neurotransmitter in the internal nervous system is acetylcholine. Acetylcholine is degraded by an enzyme called acetylcholinesterase. Okay? So that's required to end the signal. So you get neurotransmitter release, acetylcholine, it binds to the receptor, causes the effect that it has, for example, increased intestinal motility, and then it's degraded by acetylcholinesterase. Inhibitors of acetylcholinesterase are highly toxic. Prime example of these is organophosphates, which is the most widely used fertilizer in the world and the most common cause of poisoning in the world. They are also incidentally used in nerve agents in some particularly nasty conflicts. Okay, but anyway, acetylcholinesterase. This is an enzyme, and if we can find a chromogenic substrate for it, which we've used here, this will produce a colour at every point where we can see acetylcholinesterase. So this allows us to assay whether Hirschsprung's disease is present in this biopsy or not. This is effectively an assay for where these ganglion cells are, how they're distributed. So Hirschsprung's disease is due to dysfunctional innervations, dysfunctional peristalsis. The colon becomes obstructed. The diagnosis will be suspected clinically because of the obstruction, but it still requires a histological confirmation because there could be other things that are causing it. And then you do surgery to move the aganglionic region of the intestine, and then you do another histological investigation to make sure that works. So speed is absolutely essential when we're doing anything like this, when we're looking at the chromogenic substrates of enzymes, because the enzyme activity has to be preserved between taking the tissue out from the biopsy and taking it to the lab to be Right, celiac disease. Again, celiac disease is a problem with the intestinal mucosa. And you can look at the normal and abnormal small intestine, so you have a lack of villi on the intestine in celiac disease. And you can use an alkaline phosphatase method to look at that. These are HE stains, so as we were saying in the first half of the talk, you get the pink staining of the cytoplasm and the dark purplish staining of the nuclei. And you can use that to look at whether the villi are present or not. However, the other thing we can do is an alkaline phosphatase treatment, or alkaline phosphatase. It's really easy to get chromogenic substrates for alkaline phosphatase. There are lots of them used in the lab. And they generally generate a blue colour when they react with alkaline phosphatase. So you can take a sample out, you can snap freeze it, slice the sections. So snap freezing means freezing usually in liquid nitrogen, but incredibly quickly, preserves enzyme activity. And the amount of alkaline phosphatase present on the enterocyte surface gives you an indication of the structural and functional integrity of these cells. Okay. You can also use acid and alkaline phosphatases in osteoclasts and osteoblasts. So osteoblasts express alkaline phosphatase when they mature, and that's because alkaline phosphatase is required for the mineralization of bone. So you can freeze bone biopsies and you can look at alkaline phosphatase and it gives you an idea of bone health. In case of tumour diagnosis, there are more specific and simpler techniques, and something you have to remember is that for a lot of things that we've talked about, while these are quite basic techniques and they're very useful for looking at gross tissue morphology and gross features and various different components, you can look very specifically these days using immunohistochemistry, which uses antibodies which are specific for a particular protein. And I think David's going to be talking about that a lot more at 11 o'clock. Okay, so last 10 minutes or so, we're going to finish off by talking about some more different examples of special stains and examples of when they might be useful. So we've talked about these special stains already. PAS staining, remember that's periodic acid shift staining, and we use that to look at carbohydrates, primarily glycogen. Nielsen staining is used in mycobacteria identification, and enzyme histochemistry we just covered, looking at alkaline phosphatase, looking at acetylcholinesterase, and looking at ATPase and succinate dehydrogenase in muscle. So I mentioned that as well as carbohydrates, we like to look at mucins. We can also refer to these as mucopolysaccharides, glycosylated glycans, mucosubstances, or glycoconjugates. They are all glycoproteins, usually neutrally or negatively charged. You find mucus everywhere. It's really useful. Swallowing, for example. You find mucus in your lungs. The mucus in your lungs, the role is to catch bacteria and particles in your lungs, and it gets moved up through your lungs and into the back of your throat by the action of cilia. Mucus in the lungs is really important in cystic fibrosis because it's dehydrated, and so it becomes very thick. But mucus is also important in intestinal mobility as well. And actually, you get intestinal mobility problems in cystic fibrosis too. And it's all down to these mucin proteins, which are highly hydrated proteins which form lubricating layers. In any area of your body that requires any kind of lubrication, mucins are really important. So you have acid mucins, which are strongly sulfated. You find this in connective tissue, in epithelium, and you have these various different forms of mucins, the chondroitins, heparin sulfate, keratin sulfate, hyaluronosulfate. So these are sulfated acid mucins in connective tissue and epithelium. We also have weakly sulfated mucins. These are primarily found in goblet cells of the large intestine. So I'm sure you're going to cover gut mucosa in a lot of detail with David, because it's really important in colorectal cancer. So here's an example: goblet cells in the intestine, and we are staining these for weakly sulfated acid mucins. You can see that these are shown in this nice blue color. These are intestinal crypts, and these are goblet cells. So unlike acidic mucins, neutral mucins don't have acidic side groups, so we need a different sort of stain for those. And these are stained with Alcium blue. And these are sort of found in epithelial-like areas, and they're very abundant in the lining cells of gastric lining cells. Here's an example of where you might find a problem. This is metaplasia of the esophagus. This is Barrett's esophagus. And what happens in Barrett's esophagus is it's usually caused by the major risk factor is poorly controlled acid reflux over many, many years. And it results in the formation of gastric-like or intestinal-like epithelial cells in the esophagus. Okay, so this is metaplasia. And it's often regarded as a precursor or a precancerous state. So 1 in 10 cases of Barrett's esophagus leads on to esophageal cancer. It's a particularly aggressive and difficult to treat form of cancer. So looking at glycogen and mucins is really important in different tumours. The distribution of glycogen can change. For example, carcinomas of the bladder, kidney, ovary, many different ones. These are all mucins. The distribution of these and the appearance of these in tissue sections can change significantly in all these different tumours. So staining for different tissue components is really important when we're looking at tissue abnormalities, particularly in your case because it's cell pattern oncology, in different forms of tumours. As well as pathology, we can also look at the presence of foreign bodies. I showed you about asbestos bodies, but as well as by foreign bodies, I mean pathogens as well. So this is a particular stain fungi. This is Grocox methylene silver stain. Now, the thing to remember about silver staining is silver doesn't look silver on stains, it looks black. Always remember that. Okay? So silver deposits always look black. So anything that is stained by silver is black. These are fungal bodies. And this is a fungal infection of the lung. So in this stain, acid oxidation of the cell walls of the fungi, the cell walls of the fungi will contain polysaccharide. Acid oxidation will form aldehydes from that, and that will cause a reduction in, if you use alkaline silver reagent, there's a hexamine silver complex. These aldehydes will reduce that complex and result in a deposition of silver. And that is why we get these black bodies being formed here. And this shows us our fungal infection of the lung. Fungal infections of the lungs are generally quite rare in people with normal, healthy immune systems. However, they become increasingly common in people with compromised immune systems, particularly HIV. This is another example of a silver-based stain, the Mason-Fontana stain, or Masson-Fontana. And this is a stain for melanin-producing cells. Okay, so melanin is a pigment, and melanin reduces solutions of ammonial silver nitrate and results in deposition of silver where the melanin is found. However, you can also stain other components, such as some components of the intestine with this. Okay. So this is one of the textbooks which Sarah recommends. This is a snapshot of the library, I believe, the library website. So that means this book's available in the library, should be available online as well. I'm not sure whether or not there's an online reading list for this module, but my suggestion to you is if there's not a reading list on there already, email Sarah, she'll get back to you with some textbooks which will be particularly useful for this topic. There are important considerations in when we're thinking about staining for specific components. We have to think about how we fix something, what we want to demonstrate, what tissue it is, and how we control it. And this is pretty much true in any staining procedure. So the point of fixing is you want to immobilize and make your section chemically stable, right? But you want to preserve the structures that you want to look at. So you need to use a fixation method which will still allow you to see the things that you're interested in. What you want to demonstrate, that will be a consideration when you're thinking of what particular aspect of this, what questions you want to ask about tissue, what structures are you looking at, what tissues. This might affect what confounding factors you might get to know, what artifacts might you get, what things might your stain identify which are not what you're looking for. So you have to have some knowledge of what you've actually got there. And with all these things, you have to find a control, suitable control. So something in which you do not expect to see this apparent feature that you're looking for example. Right. That's a lot. Thank you for your attention. The summary of this, we've looked at the basics of why a dye is coloured. We looked at key terms, auxochromes, chromophores, chromotropes, metachromasia. We looked at examples of the chemical structures you find in many different dyes. Why they're coloured and how they bind to the tissues they bind to is all important. Hemotoxin and eosin are probably the two most important histopathological stains. We've looked at how dyes interact with tissues, the importance of the different basic tissues components in dyes, types of binding between tissues and dyes. We looked at azo dyes. We've looked at how you can have a dye that doesn't actually have a charged component, basically not a stain really, but will stain fats because it is fat soluble. We looked at the use of metachromasia, and these thiazine dyes are the ones that usually exhibit metachromasia. We looked at how we can differentially stain tissues by using tissue permeability as a differentiating factor. And we looked at local formation of a coloured substance. For example, enzyme histochemistry, such as dehydrogenase, ATPase, alkaline phosphatase, acetylcholinesterase. And we've also looked at a few different diagnostic applications, which I'm afraid is not my specialist field, so apologies if that wasn't particularly clear. Any questions you have on this, please email Sarah. She will be back next week, and I'm sure she'll be delighted to have an inbox full of emails from you guys asking for things that I didn't make sufficiently clear. Okay. Thanks for your attention. You got any questions about the chemistry of this? I'm happy to answer. Questions about the diagnostic implications, I will try and answer, but can't promise I'll be any good. Right. David's going to be along at 11 o'clock. He says precisely 11, okay, because he's got quite a lot to go through with you. But you've got a good 20 minutes to grab yourself a coffee now, okay? I'll hang around for 5 minutes in case anyone's got any questions. Thanks. That's a good question. Anyone got the attendance sheets? Okay, can I have the attendance sheet, please? Yeah. There's some people down here who haven't signed it yet. That'd be great. We'll bring coffee along and I'll have a look at each other's CVs. That all depends on you. I think not having names on them is probably a good thing because then people won't feel so. Because we did with my little two of them, so yeah, you can leave your name off it. That's probably the best thing. Just CVs. That's what you need to bring along. We're going to be doing many CVs in the next one. We'll also talk a little bit about some science and new stuff. I think Cameron said he was going to do that. I can't remember who said they were going to do that. They have such long time in the APT. I think the progression report is 5%. 5% progression report and 5% that reflective practice professional portfolio. Yeah. So together they're 10%. It used to be 10%. And you get the progression report has nothing to do with it. That's your project supervisor. They will mark that. But I will mark your reflective practice portfolio. No worries. It's Monday, isn't it? You know?
Yeah, Monday.
Yeah. Cool. That would be really bad, wouldn't it? You missed the whole weekend. Thank you. So what you do?
So good. I was going to do that.
Yeah. Yeah. Yes. I don't think it's just it's got the clear document so I think that's the point of understanding.
It's good.
Yeah. Or also, yeah, the force square. So chrome a dyne is chrome with colour, and also chrome to link it to the so apart from it, I can use a mortar. Okay, so chemotoxin uses a mortar. So chemotoxin is a mixture of unity and one of these compounds. Put it onto the cell, and then use the link between the dyne and the tissue. The dyne is an oxychrome. It's the oxychrome that looks at the tissue. So the dyne itself. Hello.
Oh, it's Cameron. It's Cameron.
It was Cameron. Right, Logan says Cameron. Good. Good. As long as we're all agreed on that. Alright, see you guys later.
Just the last 2 days I spent more money on bank card than the first 2 weeks, so it's good just from like work cash. Yeah, off my friends. I didn't have one, and now I'm hungry.
Yeah, that's why
I do that.
well, I don't have any more to do, right?
If someone, that's never gonna happen.
No, I will, because
I'm talking about there's not gonna be a point where you're not gonna have
right now.
We gotta do that.
Thing is,
The last minute decision.
it's on Tuesday.
hours in the week.
That is not called.
It's Sunday afternoon. So that's how long for a course it is.
Yeah, but I can stagger it.
Oh, I'm saying, no, my point was no, I didn't think there's going to be a point to actually have no course.
I'll have a course thing, but it's not going to be like, fuck, I have a course thing. It's going to be like,
You made a start on any of the stuff for Monday. We have to do 3. My representation is kind of like ensuring our understanding is actually translated. So we're not just going, not actually taking anything in. You have to be able to find
every single question you have, every single one. That's meant every time. That's about like 5 times. Correct.
Sounds very good. One of my favourites last year was the third week we had shown it. And it just really highlighted how much I love it. I haven't had a voice for a while, so it's really hard for me to remember what it sounds like. So when I remember this really like soft Scottish accent. I love shows.
Yeah.
Your job is to just like judge.
That right there is where people die. If we can see the screen, comes you to stage.
Anybody tell me what you guys have done in the last 2 weeks? When I look around the room, everyone just sat there. And I just thought to myself, like, come on, people.
You cannot be wrong.
It's been 2 weeks.
Even if you are wrong, she doesn't.
Anybody tell me what time it's? Everyone's just like frozen in fear.
Is this philosophical? Is there anything else? And then it's like that. As far as things that work, I let people know when they were there's no patient. You know how you're telling me like Dr. Leo would always post feedback when you saw staring. I'm just not at you, and every time I'm just sat there like this.
Did you not talk to me on the first name basis?
I'm not sure if I went scale.
Yeah, but if you say Dr. Christie, I don't know.
You know,
Yeah, she did really well.
yeah.
She had to tell parts of
It's a scale
Yeah.
or something.
Do you even work here?
Maybe even a few.
So I happened yesterday. My induction, she just looked at one of the girls and was like, Devon, and I was like, oh, girls, thanks. Look, this slide. I printed off the one for immunocytic for explicitly said there were no handouts for it. Yeah, I mean, what? That was 6 slides per page, right? Double sided. Single sided. Oh, no, actually, immunocytic.
Oh, yeah.
This one's
No, even it could be right. This is not that way.
Here are my findings of units.
Way for back.
Someone combined them.
Maybe. Everyone.
Come quickly, quite dilute.
So you go to the experiment. Who's inhibiting me?
Morning, guys. How are you all? Not working. How is everyone? Stressed? Excellent. Good. People do their best work under pressure. Ask Tim. I did 2 hours of stain theory this morning. That was quite brave of him, wasn't it, really? How did it go? Yeah, good. No, he was a bit worried about it, but it was really, really kind of him to offer to step in and cover that because I didn't want him to get behind. Obviously, if there's aspects of that you're stuck with, I believe Sarah's going to be back with us next week, so we can pick up on bits of it in the practical, because next week's practical is all really about the stuff that we've done so far. So we'll be doing some stuff around fixation, some stuff around fresh frozen versus paraffin embedding, and a little bit of stain theory stuff. But it's really just an excuse to get in the lab and actually look at some things for real, rather than just on lecture slides. Okay, so so have we got any questions about anything else? Yes. Yeah, I mean, there's a chapter in Bancroft and Gamble, which I've put online for you, which is all about theory of staining. And the other thing I would say, from the stuff last week and this week, this book is actually incredibly useful. And it's really quite easy to read. What I would say is, having read the chapter on immunocytic chemistry last night, it's all right. I've done enough immunohistochemistry to bore the living crap out of all of you. All right. I just wanted to have a check through the book, see what the book said. And what I would say is that the book is actually quite a bit out of date. In some aspects, but in terms of basic principles, it's not. Don't worry. From a research perspective, it's perhaps a little out of date. From a clinical perspective, it's probably absolutely fine for what you need to know. So, yeah, don't worry to worry. Let's not send that to export, because we're having real problems with this panoply thing. I'm trying to record all the lectures for you. However, what happens is that when, at the end of the recording, it has to upload, and it's been every lecture theatre I've been in so far this term, it just failed to upload, which is really, really annoying. Anyway, we'll see what we can do. Okay. One thing I will look up is, this will make no sense, so just bear with me. Have you got any questions about the essays, guys, actually, while I'm studying? I did say I would happily talk to you about it during lectures. Anyone started? Anyone prepared to admit that they've started? Right. Anyone that hasn't started, why not? Like, give us a list. That's only like a page and a half or something. That's easy peasy. I didn't ask you to moan at me about all the problems. Okay. You've got to remember it's very early in the term, right? And the progress reports, they do come around very quickly, but they are what they are. They're 5% of the module. They're really just to show that you've engaged with the project and that you've got some idea of what you're supposed to be doing. You're shaking. No? Do you want me to give you a project? I'll give you a project now.
Oh, I have one. I just went into the project and was like, Well, I mean, there is. You've got to remember there is health and safety, and it is there to protect you at the end of the day. And you can't get your lab passes until obviously everyone's had to have their inductions and the risk assessments have to be signed off, and you need to look at them so that you know the stuff, you know the dangers of the stuff that you're working with. Really. And in fairness, I'm not trying to stick up for anyone. I can feel your frustration, but that doesn't hinder you from doing the progress report, which should take you, I don't know, a couple of hours. Okay. All right. So what's going to be the next thing after the progress report? Cool. As I said, as you're going along, you probably will have some questions about the essay. Just come along on Friday morning and ask them, and I'll happily talk about stuff. Yeah. It's much easier to do that than to have to try and respond to like 30 different emails about things. I can't write 30 essays on how to write an essay in an email, so it's much easier to talk to all of you about it. Yeah. Okay. Ooh, I don't know. I might say about the magic triangle, and I'm looking at the project students behind me. The structuring of the essay. You see, this is where people say, "How should I structure my essay?" And I thought, well, I can't answer that for you. And it sounds like a cop-out answer. It's not. But learning to structure an essay well is actually part of the challenge and part of the work you get marked for. Now, if you've read the assessment brief, it gives you some clues as to what kind of things to include. But the topic that you choose will dictate the balance between them and what aspects that you focus on. So, to take for example, if you wanted to write an essay around cervical cancer, as I said last time, you would probably focus more heavily on screening, vaccination, things like that. Yeah. If you were to write an essay on colorectal cancer, you might go one of several ways, but you may focus on the influence of diet or microbiome, something like that. Yeah? If you're focusing on lung cancer, what you could do, as I'll tell you in the lung lecture, and you'll find out this afternoon, is that nearly 7,000 people a year in the UK die from lung cancer that never smoke. That's quite an interesting topic. Yeah, what I'm saying is that any particular disease that you choose, you will find that there is some contentious issue around it somewhere. And what you want to do with your essay is being able to get into the scientific literature, the recent scientific literature, what is happening in that disease area research at the moment, or over the last couple of years. And the closer you can push that. So I can't tell you how to structure your essay because it depends which disease you pick. I mean, I could have picked a disease and told you all to write about one particular thing, but that's not great, because then you probably wouldn't like it very much. You probably won't like writing the essay very much anyway, but at least you get to choose the topic. And hopefully it'll be something that you're interested in. Right, I'm just going to look up a picture which will make no sense whatsoever to most of you at the moment, but I hope will come in useful early, because I realised after I'd gone home last night, this wasn't actually in the PowerPoint.
I'll control.
Right. Okay. Huh?
I'll control and scroll. Click the mouse forward.
Yeah, okay.
No, okay.
Or you can click
Don't worry. I'm going to just see if I can dump it into this PowerPoint at an appropriate place. I could just put it in an inappropriate place, as long as I know it's there somewhere. I had a PowerPoint with somewhere in the region of 95 slides to an hour. So I've trimmed it down to 60 odd. Let's just dump it again. Sorry about this. Hooray! Look at that. That wasn't that difficult, was it? Right, cool. So, without further ado, get on with it. Let's see if we can record this. Okay, so last week we talked about fixation, processing of tissue, stabilization, that kind of stuff. Now, obviously, this is the kind of technical aspects of cellular pathology, so this is the kind of nuts and bolts of what goes on in the cell pathway. We will be coming back to a lot of these topics as we go through the applied lectures, and hopefully it will kind of connect up a little bit. I'll give you some examples as we go through today. So Tim's talked to you about stain theory this morning. That used to be actually, I think, about 4 or 5 hours of lectures, and we've actually condensed it down. So I hope that's worked okay. But just so that you get, there's a balance here between the fact that we live in a world where you can just go and look something up. Yeah, you can just go and look up how a particular stain works and what affects it. If you pick up one of the big staining books, you can find it, which is what someone would do in. I keep pointing this out, sorry. Because you look that's what you would do if you're in a lab and you were stuck. However, understanding some of the principles underlying stain theory and histochemistry is important for you to be able to understand what's written in the book. Yeah? Okay? That sounds fair? So, use of stains is obviously very widespread in cellular pathology for illustrating things in tissue, and over the next couple of practical classes, we're going to look at a few different examples of stains and why specific stains are used for specific things. And in lectures, we will look at how specific techniques are used to diagnose diseases in certain tissues. Okay, and so your choice of stain for your tissue depends on what you're looking for. But in the last 20 years or so, a technique that has become very widely used within cellular pathology is immunocytic chemistry. Although I would tend to err on the side of calling it immunohistochemistry, but this is a bit of a cover-all term, and I'll try and explain why as we go through. Now, I've been through Sarah's PowerPoint. I've said I've chopped it down, but I have no idea how it's going to animate, so if anything weird happens and I look shocked, that's why. Does anyone here know much about immunohistochemistry? Has anyone ever done any? You've done some? Yeah? What, research-wise or routine lab-wise?
Yeah, I did some on my placement.
Cool. What were you looking at?
I was studying spinal cord samples in mice.
Okay. And using a range of antibodies or with a single?
I did a series of DAB stainings and using antibodies for vitamin D receptor and prokinase.
Okay, cool. Oh, that sounds interesting. How about yourself? Ah, nice. Did it work? Cool. Anyone else? Yeah, what have you done? Cancer stem cell. Who are you working with? Why is it open? Did you get to work okay? Well, and I'm not picking on you guys. It's nice that some of you have some experience. It's great because you can come up and deliver those bits of lecture now. My experience with immunohistochemistry is all research-based. I've done a lot of this, but all from a research perspective. And we will talk about that later in the module. Certainly when Sarah and I do our research seminars with you, some of that will be around looking at tissue specimens, looking at immunohistochemistry, and the way that you store that data, the way you interpret that data. So I have that kind of bias on this topic. However, it's also a very important technique in diagnostic pathology. Okay, and I'll try and give you some examples. It can't be an exhaustive list in the next hour. I'm going to go through the principles of how this technique works for those of you who aren't familiar with it, and it'll be a refresher for those that are, including myself, hopefully, and I'll try not to trip over it too much. But get the basic principles, and I'll give you some ideas of how and why it's used. What I will say is that I'm not going to be able to cram all that much into an hour, when you have the other lectures, so for instance, when we do kidney lecture, I'm going to talk to you about immunostaining of kidney sections for diagnostic purposes. When we do the lung lecture, I'm going to talk to you about being able to distinguish mesothelioma from metastasis, etc. Lots of times during, and when you do the breast cancer lecture, Sarah will talk to you about doing ER, PR, HER2 staining for prognosis and treatment in breast cancer. So the principles of today, but the application of this technique is going to crop up in a lot of different lectures, a little bit like I just said the stain theory would. Bonus points for anyone that can tell me what that is. It is a stain section. Yay! I wish I'd brought chocolate. It's actually a carcinoid tumour. And carcinoid tumours, I remember the first time I saw one down the microscope, some wag of a pathologist sneaked one into my batch of 100 colon cancers that I was studying. And this is actually a gut tumour. You can see the gut epithelium there, lots of inflammation. And I was like, what the hell is that? But anyway, yes, it's stained brown and blue. Now, just to keep this really simple, the brown stain is our immunostain, right? And I'm going to go through the principles of how that's worked. But that's what we're looking at here. And here, what we've done is, if we just use an immunostain on the tissue, remember we've got clear, colourless tissue. It's going to be quite difficult to interpret. So this one, like most of the ones that I've ever done, has been counterstained with hematoxylin, which, as you know, stains up the nuclei and allows us to get some handle on how this brown staining fits with the structure of the tissue. And I think this is a bit of thyroid actually staining up for something. But anyway, let's just example. I'm going to whiz through this bit, really, this antibodies bit, because you only need to know a little bit about the different antibodies. And the only ones you're likely to encounter immunostaining are IgG and IgM plus immunoglobulins. Talk about the difference between monoclonal and polyclonal antibodies. And if I feel we have time, we can maybe just chat, because I'm not sure how much is in the slides, around the advantages and disadvantages of each. Because it may not be immediately obvious as to why you may choose one over the other. Current methods, validation. What I would say, let's sit you all here. Validation, from a clinical perspective, is incredibly important, and I know that it's something that Sarah likes to write about. Now you know why I'm asking you to turn up on Friday. It's only because we get really low with it. So yeah, validation is a very important step, because if I'm going to be looking, like these guys have done on placement, etc., if I'm going to be looking for a specific protein marker in my tumour samples that I'm interested in researching, when I get a new antibody, I can't just expect it to work on the tissue. I have to go through a process of, one, optimizing the protocol to get it to stain. But not only that, I have to be certain that that stain is real and correct. And there's a number of steps, a number of controls, etc., that I would carry out in order to be certain of that. Because if I want to publish that in a scientific paper, I have to convince a reviewer, unless that technique's been published many, many times with that antibody, I have to convince a reviewer that my staining is real and not artifactual. Now, from a clinical perspective, if you're going to use this for diagnosing patients, you also have to be absolutely certain that what you're doing is giving the correct result and your interpretation of it is correct. And that's the validation process. So it is very important, but we will, again, be skimming through it a little bit today. But I would recommend you have a little read about that and its application to clinical practice. And then this, I've chopped a lot of slides out of this because we're simply not going to have time for it. It's the use of immunocytochemistry in diagnostic cellular pathology. I'll give you a couple of examples of the types of things, but as I said, it's going to crop up in lots of different lectures. I know it crops up in probably most of my applied cell path lectures, so don't worry too much about that. And if we've got time, I'm going to talk a little bit about quantitation because quantifying immunostaining is actually quite tricky. Right. Right. Okay. Where you can have a debate about the use of these terms. Okay. Obviously, cytology, cells, histology, tissues. Yeah, is one difference between them. You do see them interchanged a little bit. I tend to use this one if I'm staining tissue, but people sometimes use this if they're using immunofluorescence. Don't worry, I'll explain immunofluorescence in a minute. But essentially, what is this technique? It's using antibodies with labels, and we'll look at how those labels are attached, to stain components of cells in cytological or histological samples. Okay? And so it exploits the unique ability of antibodies to bind to antigens, but very, very specific antigens. Now, you've all done enough immunology at level 2 to kind of know what antibodies are. Yeah? Please tell me how. Yeah, everyone knows what an antibody is. What kind of cells make antibodies? That's good enough. That'll do. That's all you need to know. Great. We'll get on to the goats later. Yeah, okay. I had a 2-year research study that keeled over for about a year simply because the company that made the antibodies refused to admit that the goat had died. All makes sense to me. Yeah, so antibodies produced by B cells in response to antigen. Okay, antigen can be virtually anything that an antibody is raised against. There's several different classes of immunoglobulins, and as I said, there's a whole bunch of them. But for the sake of argument, the only ones you're really likely to encounter using this technique are IgG and IgM, and although they're quite different in their structure, the way they behave in the laboratory is largely similar. So there's not a great deal to be gained from knowing the distinction between them, but it's worth having an awareness that there are different forms of immunoglobulin. So IgG, sort of classic structure. So we've got the heavy chains and the light chains. We've got our antigen binding sites at the end here. This is the variable region, and here we just have the bonding together chain. Okay? So we all know the basic structure of an antibody, and we also know that this bit here is the business end that has the binding site that sticks to an antigen, which if it's sticking to a protein is a region normally around about 20 to 30 amino acids long. Okay, and it's quite a small part of the protein. When you consider the average protein is probably about, I don't know, what's the average protein? About 700 amino acids. Average gene is about 2 kilodoses, so yeah, divide by 3. But then something like insulin is what? Insulin's only what, 30 something amino acids? It's tiny. Come on, guys, I thought you did science here. This is help me out. You need to read. You need to read. Stop watching Bake Off. Right? My missus made me watch the pie thing last night. I must admit, I did feel very hungry looking at some of those pies. But I'm not a Bake Off fan. I'll pretend to be a Bake Off fan just so we can, develop some kind of rapport and camaraderie. Which means if you'd like to bring in baked goods on Friday morning, that's cool. Yeah, good with that. Okay, this is an IgM, and what you're seeing here is the same structure as an IgG, but it's repeated, so you've got 5 of them. And it's worth knowing that if you're using IgM for staining, or we don't just use antibodies for staining in the lab, we use them for doing other things as well. And people working in research science use antibodies a lot, and we are basically just exploiting that ability to specifically stick to a particular, not just to a particular protein, but to a particular region of the protein. This is IgA, and it's kind of like 2 stuck end to end with a J chain in the middle. You're not really likely to encounter that. Now, it's got some lovely graphics here. So within our antigen, which could be a specific protein, we have certain epitopes. certain epitopes that the antibody will stick to, and the antibody has what we call a paratope, although I very, very rarely hear that term used. Epitopes, yeah. So an epitope is a single antigenic determinant, and so basically it's a very small region of our antigen that the antibody interacts with and binds to. And the paratope is part of the antibody molecule which makes contact with that and binds to it. Yeah, okay. These terms I quite often get a bit muddled with, but they're very important when it comes to validation. Okay. Top tip. What did we say about validation? Yeah. Please note carefully. Specificity and sensitivity. If you're going to try and convince the Health Service or the National Institute for Clinical Excellence or whoever that your new antibody-based technique is the way forward for distinguishing between two types of cancer, which otherwise are quite difficult to distinguish, you are going to have to convince them of both the specificity and sensitivity of your technique. Now, this is slightly out of context here, but specificity means what are the chances of that antibody binding to something that's not the thing you're interested in? Yeah? So what are the chances of a false positive? Okay, that makes sense? Yeah? How specific is it? Is it absolutely 100% certainly only going to bind to the bit of protein that we're interested in? Or is there a chance that some of it might bind somewhere else? And you can actually measure that and calculate it. Okay, you can calculate with regards to a particular disease, if you've got samples from 100 patients who you know have that disease, and you stain them all, and 93% of them stain positively, then you've got 93% specificity. Yeah? In that instance. But the other thing that comes in is sensitivity. How sensitive is your technique? And I said that specificity is about false positives, sensitivity is about false negatives. Yeah, if your technique is not sensitive enough to detect the protein and comes back negative, even though it's actually there. Okay, you know what I mean? And the same approach again, you would get a range of known samples, you would stain them, and you would say if some of them come back negative, then you can calculate the sensitivity. The way Sarah's put these terms in here, so specificity is the ability to recognize only one epitope. Yeah, but I'm actually applying this to the validation process for you. Affinity is the binding strength, and the sensitivity is the amount of antigen that can be detected by the specific technique. So what kind of level of detection do we have? And that is really, really important. I mean, a lot of things, if you're looking for estrogen receptor, HER2 in breast cancer, the breast cancers that overexpress those, they make bucket loads of those receptors. So you don't need massive sensitivity because it's really easy to detect. But some things, I'm trying to think of an example, I might think of one before the end, are present in the tissue or the sample at a very low abundance. And so you need very good sensitivity in order to be able to detect them. But we also can use amplification steps in our staining, which I will talk to you about as we go on. Wow, we're supposed to be halfway through. This is going really well. Too much baked in, chat. Stop that. I'll have no more of that. Okay. The other terms that are really important for you to understand, though, are the difference between polyclonal and monoclonal antibodies. Now, have you encountered these terms in lectures before? I don't expect you to know the answer, but have you encountered these before? Has everyone encountered them before? Has anyone never ever heard of those words ever in their entire life? Come on, own up, guys. No? Right. That's really good. How many of you would feel 100% confident to stand up in the middle of the room and give a definition of each? Few, less of you. Okay, that's great. That means we can talk about it. I chopped a lot of these slides out, but the version you've got on Blackboard, I think, is the original version of this lecture. But monoclonal antibodies derived 1975, Kohler and Milstein. Before that, we only used polyclonals. Now then, I'm going to try and explain this in a fairly simple way. Right, how do we make antibodies? This is something that's not really covered in these PowerPoints. If I want to make an antibody to a particular protein, what do we do? A human protein. I want to make an antibody against a human growth factor receptor because no one has really made a good antibody to that human growth factor receptor. How do I do it? So I isolate my protein first, yeah? The whole protein or just a bit of it? Well, it depends, and it's a tough question to answer that because we've got to think about what we want our antibodies to do, and this is where we come down to this debate about the advantages and disadvantages of polyclonals versus monoclonals as well. So let's just say, right, I isolate my receptor protein. I isolate a whole bunch of it, and then I put it in a syringe. I go next door and grab their pet rabbit. Rabbits make good antibodies. They do. And I inject my protein, my human protein, into the rabbit. What happens? My neighbour comes around and kicks the shit out of me.
It's going to generate antibodies.
Absolutely. The rabbit, because the human protein is foreign to the rabbit. Now, there's niceties in here that it's very difficult to provoke an immune response to certain things because some receptors and things, some proteins, are very highly conserved across species. So there's some proteins I could inject into the rabbit and it would not have an immune response. And the companies that do this, there's a nice section on it in this book actually about how they make antibodies. You sometimes have to kind of do some quite unpleasant things and inject the rabbit with other things as well in order to try and kick off its immune system so that as part of that immune response, it will react to your protein. But the basic principle is the same. You inject the protein into the rabbit has an immune response to that, which means the serum in the rabbit's blood, yeah, if you were to take some of the blood from the rabbit in the dead of night, and isolate the serum, that serum would contain antibodies, and in amongst that will be antibodies, hopefully a large amount of antibodies to your protein of interest. Okay? But those antibodies are not derived from a single clone of B cells because it's mounted an immune response against the whole protein. There will be antibodies that recognize one bit of the protein, there will be antibodies that recognize another bit of the protein, there will be antibodies that recognize a bit in the middle. Okay? So there'll be a whole mixture, and they're all derived from different clones of B cells. You all know about B cell clonal expansion. You probably know more about it than I do. Clonal selection, clonal expansion. So that is a polyclonal antibody, okay? Because it's come from multiple clones of B cells. Now, there are advantages to those antibodies, as you'll see, because one of the things that we struggle with using antibodies to stain tissues is, remember that fixation stuff last week? And we've cross-linked all our proteins together? Well, that can obscure the antibody binding sites, the epitopes that we're looking for. They can be all cross-linked up, and we can't actually get at them. So if we have an antibody that only recognizes one specific bit of the protein, and it can't access that bit of the protein, we're a bit stuck. And in that case, a polyclone will be really useful because it will recognize all the different bits of the protein, but it will be less specific because there will be a good chance that those antibodies can recognize things that off target. Yeah? So it will be less specific, but it may be quite sensitive. Can you start to see where this trade-off is coming here? Monoclonal antibodies are made in a different way. What essentially happens is you inject your mouse, in this case, and you can isolate the serum, and as I said, it will have a mixture of antibodies, in this case, antibodies 1, 2, 3, and 4, which will recognize the antigen, but in different places. And that's what I've just described to you. But to make a a monoclonal antibody, you isolate the spleen cells. Immune cells do not like living outside the body for very long. Okay, they're not that hardy. So in order to make them live for a long time, they are hybridized with myeloma cells. So it's basically fusion of two cells together. So you fuse your plasma cells, your B cells, with a tumor cell, which makes what we call a hybridoma. Okay, and we can grow those hybridomas in culture, and you can separate them out, that you make separate colonies. So those separate colonies will, once you've figured out which one's which, each colony of cells growing in a separate dish or separate flask will make only one antibody, which will recognize only one epitope on your antigen. Okay? And that, therefore, is called a monoclonal antibody. And what does that do? Well, in terms of specificity, it really ramps it up, doesn't it? Quite obviously, it's only recognizing one particular part of our protein. So the chances of it being very specific are good, but we may be compromising sensitivity. Is that clear? Because if you're not familiar with this, I know that's quite a lot to take in. Does anyone want me to go through it again? Do you have any questions on that? Are we fairly happy we understand the difference between monoclonals and polyclonals? And have you got some handle on why you may want to use one versus the other? Yeah? Most antibodies that are commercially available, and these are the ones used, the ones used in diagnostic labs are largely commercially available antibodies as well, and are derived from mice. So you have mouse monoclonals, rabbit, I wasn't kidding about the rabbit, polyclonals, donkey, horse. They're the most common ones that are used. And it's important to know those species, because when we come to the secondary antibodies, we need to know a little bit about those. So if everyone's happy with that, we'll move on. So monoclonal recognise one epitope on an antigen, and they all have the same affinity and specificity. Polyclonals recognise several different epitopes, and they vary in their specificity between the different antibodies within the polyclonal. So as I said, from a practical perspective, there are times when a polyclonal can be useful. So current methods used, right? The most commonly used method in immunohistochemistry, which is where I should hand over to my project student, is the ABC method. And we've kind of skipped a bit here, maybe. Because what we want to do, we've got our primary antibody here in red, bound to our epitope. That's fine, that's great, except we cannot see that down the microscope. Right? We can't see it. Without wanting to sort of jumble the order of the lecture too much, we can tag a label onto that primary antibody. We can put a fluorescent label on it, or we can put some kind of chromogenic label on it. Why would we not want to do that? Well, any ideas? Why would we not just want to directly label our primary antibody? It does happen to some stuff. If it was in the research laboratory, I would not want a directly labeled primary antibody, because I will want to use that antibody for immunohistochemistry. I want to use that antibody for immunofluorescence. I want to use that antibody for Western blotting. I would also want to use that antibody for things like flow cytometry. That's because antibodies are really expensive, yeah? Especially primary, really expensive. So I will want an antibody I can use for a range of things and be able to label differently depending on the application. In the diagnostic lab, that maybe is slightly less of a consideration, because they will tend to have an antibody for one particular use. So in order to be able to detect it, we're going to use a second antibody. Okay? And that second antibody doesn't recognize our protein of interest, it recognizes our primary antibody. Now then, I know this can be a bit confusing, but it's actually really, really quite simple. So I've made my primary antibody in a mouse. mouse monoclonal. Okay? If I get a load of my mouse monoclonal antibody and go back through the fence next door and grab the rabbit and inject my mouse monoclonal IgG into that rabbit, what's the rabbit going to do? Apart from squeal a little, maybe wriggle. Sorry? Yeah, the rabbit is going to mount an immune response towards the mouse IgG, which means that the rabbit serum will then have anti-mouse antibodies in it. Yeah? With me? So that's my second antibody. And the reason I'm kind of being a little bit, hopefully not too patronising about this, is that the terminology does sometimes pop people, because I'll say to someone, what's the antibody you're using? They go, oh, it's rabbit. And what is the primary or the secondary? And I go, no, it's rabbit. It's like, okay, well, your primary antibody is mouse, your second antibody is raised with a rabbit, so you'll have rabbit anti-mouse. Yeah, but you can also have goat anti-rabbit. Yeah, you can have horse anti-goat. And if you're using antibodies, you need to understand this principle in order to get that terminology and not be confused by it. But this, you said, what about the mouse one? Well, the mouse is mouse anti-human because it's designed to, it's raised to detect a human protein. So we use a secondary antibody and apply our label to that, which gives us more flexibility. But in this case, the ABC method, what it allows us to do is to amplify that signal. So if I'd have just in this case, we're going to use biotin, right? Biotin is our label. If we just biotinylate our primary antibody, we get one biotin molecule, one binding site on that antibody. What the ABC method allows us to do is if we have a biotinylated secondary, we can use something called avidin, streptavidin, which has 4 biotin binding sites. And each of these biotins has got a little enzyme stuck to it. Anyone here like horseradish? Does anyone here like food? You like food? There's going to be a lot of food chat over the next few months, guys. Get used to it. Because I don't eat breakfast, I get obsessed with talking about food when it gets this time of day. The enzyme on most of these, there's a few that are used. The most commonly used are horseradish peroxidase and alkaline phosphatase. But I'm probably going to talk mostly about horseradish peroxidase today because it's used far more than any other. So what it means is that empty biotin binding site. Can you guys see it there? Sorry, I forget to look at the screen. That empty biotin binding site on the abdomen binds to our second antibody, and rather than just having one enzyme label, it's now got 3. So we've tripled the potential signal from that binding. And we use those enzymes to create a colour change reaction, which is very nicely making that brown colour. So we're going to go through this with Sarah, but we're going from the bottom. I'm going to talk about antigen retrieval in a moment. We won't worry too much about that. You can come back to this slide and it will make more sense afterwards. We bind our primary antibody to our antigen of interest, and we normally incubate that overnight in the fridge. We wash, because obviously not all the antibodies stick to it. We wash it, normally with something like PBS. We wash. We add our biotinylated secondary antibody. That's the biotin in blue there. We leave that on for half an hour or an hour, which is raised against the species of the primary antibodies. This will be our rabbit anti-mouse in this instance. We then add this ABC reagent. The ABC stands for avidin biotin complex. Okay? And you can buy this as a kit. You can make it yourself, or you can just buy the kit. And as I said, the avidin pockets bind with biotin, but we've already got 3 biotins with our enzyme label, in this case peroxidase, horseradish peroxidase, and this is an amplification step. Okay, so we've now got 3 enzyme molecules that are antibody binding rather than 1. And again, there's washes between each step to wash off any that isn't stuck. If you don't wash it properly, everything will go brown when you put the substrate on. And then the final step is you add a substrate called DAB, which you mentioned, you've done this earlier. And DAB is a kind of clear, colourless liquid. It's actually quite toxic, but so we've got a risk assessment for that. When you put it on there, the peroxidase enzyme turns it into a brown coloration, and it literally takes a few minutes to do that. That's the basis of the ABC method. I'm going to sort of accelerate through this a little bit because this is when I was talking. I've already mentioned this. This is the process of cross-linking. When we fix our samples in formalin, we form cross-links between the proteins, which means that our epitope may not be available for the antibody to bind to. So before we do add any antibodies to it, I'm sorry this jumps backwards and forwards a bit, is we have to do something called antigen retrieval, which is a bit of a dark art, really. Antigen retrieval, and it's part of your protocol optimization. So if you're using an antibody on a sample type that hasn't really been done before, you may have to optimize this particular step. The most commonly used antigen retrieval method is heat-induced epitope retrieval, HIER. You can do that by putting it in a water bath, you can do it by putting it in a microwave oven, and you can do it by putting it in a pressure cooker or an autoclave to raise the temperature. And basically, cut a long story short, you tend to put your slides, having de-waxed them, etc. You will put them in citrate buffer, and you will heat them for a certain length of time, which then you would have optimized that process to retrieve your epitope of interest. You can use enzyme digestion, etc., but by far the most commonly used method is heat-induced epitope retrieval. So the types of labels we can use fluorescent labels, which is why I stuck that slide in at the end. This can be direct, so this is our primary antibody sticking to our protein with a fluorescent tag on it. But as I said, in a research setting, I'm going to want to use that primary antibody for lots of things. So I will use the primary antibody and then use my rabbit anti-mouse with a fluorescent label on it. So that's either direct or indirect fluorescence. In a diagnostic lab, in a hospital lab, direct fluorescence is used for certain things, and because it's quick, it has less steps in it, it's quick and easy, and they're only going to use the antibody for that technique. And we could talk for ages about how you actually stick a fluorescent tag onto an antibody, but just don't read about it. There's just some examples of immunofluorescent staining. This will crop up again when we have kidney lecture. Week after next, I think it is. It's certainly coming up in the next couple of weeks. This is a staining of glomerulus for immunoglobulin deposits, which allows you to diagnose kidney disease. And I think this is it's a bit thyroid, actually. Staining of immunofluorescence. Did you have fun making fun of Tim with the what's that tissue? Yeah. No? Okay. Cool. Same thing applies to chromogenic labels. You can directly label, but as I said, if you directly label with your horseradish peroxidase enzyme, for example, you've got one. Whereas if you put it on a secondary antibody with the ABC method, you've got the opportunity to amplify up that signal. There are other ways of doing signal amplification as well, but we haven't got time to talk about all of them, unfortunately. Okay. What's happening here? I'm going to mention something important here. I can't remember if it's in the slides or where it is, but we've got a primary antibody or secondary antibody on there, and here's our ABC. So we've got our enzymes on there, and basically this is just showing that the reaction with DAB from colourless to oxidized, which then gives you that brown colouring. Now then, there's another step, which is quite important, because tissues have endogenous peroxidases in the tissue. Now, even when we fix tissues in formalin, it doesn't necessarily mean that it completely inactivates all of the enzyme activity in that tissue. So if we were to just use this method, if we were to get some of this DAB stuff and put it on a tissue section, an aqueous tissue section, with no antibodies on it at all, it would go brown, because the enzymes that are still present in the tissue are capable of carrying out this reaction and carrying out the colour change. So when we stain samples for immunohistochemistry using this method, we have to inhibit those endogenous enzymes. And to do that, we just soak them for a few minutes in hydrogen peroxide. Yeah, and we can do our hair at the same time. We soak them in hydrogen peroxide, and what that does is it basically floods those enzymes that they then lose their activity, so that when we do our antibody steps, they will no longer cause this, because otherwise we're going to end up with non-specific staining, non-specific coloration. And there's a few things that can lead to non-specific color staining, but so these enzyme labels are usually peroxidase or alkaline phosphatase, substrates for peroxidase, and you can use phosphatase for alkaline phosphatase. And you can do dual colour staining as well, so you can actually use both. So you can stain with one antibody and bind alkaline phosphatase to it. You can stain with another antibody and make peroxidase to it, and you can get two colours in one piece of tissue. Getting that to work well is really quite tricky, and it's probably something that's more likely to have research-based applications than you would find in a diagnostic laboratory. This is just diaminobenzidine tetrahydrochloride. It's quite toxic. You can kind of tell that from its name, can't you? Anything that's got a name like that, you know, anything that's got like benzidine in there, oh yeah, there's definitely, you know, a little bit of nastiness. It's not a very nice chemical, but very, very useful. And basically, it's just converted by the enzyme from a colourless chemical to a coloured product. Right, let's have a few minutes about validation. I've talked about this at the start. I would ask you to read about it, because it is important, and it's an important consideration. It's an important consideration for you guys, because if you want to go on and work in a hospital lab, you'll need to know how things are validated, or it'll be useful for you to know how antibodies are validated for use. If you go on to work in research, you will want to know a little bit about how to validate your own antibodies in order to get a reviewer of your superb research paper you've written to believe you that your stain is right. So the principles of this are have quite broad application anywhere that you might be using antibodies in your work. Oh, that's nice. Right, okay. Oh wow, more spinny things. We did this last week. Oh, what have we got here? Optimal antibody concentrations. Yeah, you have to work out the optimal concentration, which means you need to do the titration of your antibody at different concentrations on your tissues. There's bits here on specificity, specimen preparation, so there'll be a bit here on heat-induced epitope retrieval. So this is DACO. DACO are kind of like you'd know DACO, they're very expensive. They're like the Waitrose of antibody shops. Yeah, in fact, they're posher than Waitrose. They're better than that. I used to buy most of my antibodies in Aldi and pay the price for that. Actually, Aldi's great. Let's not knock any supermarkets. Apart from all of them, because obviously all these are killing off local shops. But apart from that, DACO are like the sort of gold standard people for antibodies, certainly within a diagnostic lab. If any of you ever go and have any experience working in a histopath lab in a hospital, you will see lots of boxes of stuff in the fridge with DACO. They make superb antibodies, but you pay a premium for them. This is the data sheet for a monoclonal mouse antibody to human P10. P10, Sarah will talk to you about in the prostate cancer lecture. It's an important prognostic marker for cancers. She's put this in because she's used it for research purposes. But on this data sheet, they're giving you the clone number of the particular mouse hybridoma that was used. They will give you some indication of what dilution to use the antibody at for immunohistochemistry, depending on what detection system you're using. They'll give you some data on the specificity. You'll notice there's some cell lines, some breast cancer cell lines in here that they've used as controls. And they will tell you a little bit about how to do the antigen retrieval. But you may still need, you can go from this, but you may still need to work it up yourself because in the conditions in your laboratory. What on earth is that? Okay. Oh, thanks, sir. Cheers. PH9. Remember that. Yeah. hopefully get to mention this in a minute, but it's giving you clues to what the staining pattern is here. If you go on the website, they'll probably have some pictures of controls. What it's saying is that it should be cytoplasmic and or nuclear stain. And localization is quite important. So when validating an antibody, we need positive and negative controls. So you're going to stain something that you know should have that protein in. That's your positive control. And you can stain something that you think shouldn't have that protein in. It's a negative control. Other things you can do, you can use a blocking peptide. Blocking peptide is the peptide that is injected into the mouse to cause the immune reaction, right? You can buy that from the company that will sell you the antibody. What you do is you get a little Eppendorf tube, and you put some antibody in, and you put the peptide in, and you give it a flick, and you leave it for half an hour, and all of the antibody that's in that tube, because you put loads of peptide in, should bind to the blocking peptide. Okay? You then put some of that mixture onto your sample, onto your slide, your tissue slide, and it should be negative if you haven't got any other non-specific coloration. They're generally not completely negative, but they'll be much, much weaker than if you just stained it with the antibody. And that proves specificity, okay? Because the issues you can get is your second antibody might stick to something a little bit, you might not be washing it properly, you might end up with a bit of dab staining, you might not be blocking your endogenous peroxidases properly, all of those things. If you do the blocking peptide experiment and it's still going brown, you're going to ask yourself some serious questions because you've blocked the ability of your primary antibody to stick to the target. Is that clear? Is anyone not clear on that? Okay, really important. When I get around to finishing writing my notch paper, you can read the supplementary information in that paper will bore you absolutely senseless. So if you're having trouble sleeping, keep a copy by the bedside, because we have had to prove beyond any doubt that the staining we're seeing in the tissue is real, is the opposite of what everyone's expecting to see, but we've had to do so many controls to prove it, and we might actually talk to you about it in the research seminar session. Oh, okay, I'm just going to let you look at this in your own time, but this is obviously just a flow chart to work through. This is actually for Western blotting, I think, yeah, so it's Western blot there. It's basically the questions you would ask yourself to confirm whether your antibody staining is correct or not. This is blocking peptide use, I just said, so you stain with the antibody, you get this. If you stain a serial section with antibody that's been blocked, you can still see there's a faint bit of colouring in there, but it's blocked the vast majority of antibody binding, and that tells you that the staining should be specific. That is the worst graphical representation of the Western blot I've ever seen. There's some of Sarah's Westerns as well. It's quite a nice Western. Western blotting you can use to confirm that your antibody is working properly. Basically, you can run the proteins out on a gel and then transfer them onto a membrane. You can then immunostain that membrane. Now, Western blotting is very like immunohistochemistry in terms of principle, except you're using proteins on a membrane rather than a tissue slide. You use primary antibody, you wash, you use secondary antibody, you wash, you use a detection agent. Yeah, in this case, it's normally a luminol, which is a luminescent agent. In this case, is optimization, antigen retrieval, dilution of your antibody, as I said, incubation time, incubation temperature. Same with the secondary antibody. And but optimization is an important process. Optimization and validation, very important diagnostically, but also in research. What to look for? Subcellular localization. So we've got nuclear, cytoplasmic, and membrane-bound antigen in these three examples. This is quite interesting. I could tell you backstory to this, but I don't have time. This is an invasive front of a breast cancer sample, which I actually remember telling Sarah to look at. This is some normal breast duct here. This is our breast cancer here. And this is what you see is the breast cancer cells away from this invasive front are not staining as darkly as the ones at the invasive front. And that, in research terms, tells us something that that protein might be involved in tumor invasion because we actually see it localized within the tissue. So localization within the cells is important. localization within the tissue is important. Don't know what that is. Don't care what that is. Interpretation, we can score them. This is HER2. If you have breast cancer, your sample will be analysed for ER, PR, and HER2. Don't worry, Sarah's going to go through this in detail in her lecture, and we'll also do it in the practical case study. If from the immunohistochemistry, remember this is lectin receptor, so it shouldn't be in the membranes. If you come back with a score of 3, it will be you will receive receptin. 2, it will go off for in situ hybridization. See if you've got I'm not going to talk too much about this. So these are all examples that are going to crop up in the lectures. What I will finish with is immunofluorescence. I did just want to share this with you. If you look, it talks about something called fluorescein isothiocyanate. That's a little bit dated. These days, what we use is secondary antibodies tagged with these conjugates. And what this allows is, what we have is these epitopes, these conjugates, which emit light at different wavelengths, and emission spectra. And we tend to use 488 for green, and I also tend to use personally 594 red because they're quite separate. But what that allows me to do is it allows me to fluorescently stain cells with 2 markers at the same time using different colours and overlay them. And you would have seen those images in journal papers where people are actually looking at the localization of molecules in cells and you see different coloured staining. And that's exactly how they work. And I just wanted to show you that quickly to give you this idea that you need some that sit separate, that they don't overlap. If they overlap, you'll get bleed through when you try and image them. Okay, I'm sorry that was a bit of a rush, but I hope you've at least got how this works, and then we can actually look at how to use it in practice in lots and lots of other lectures, okay? Thanks, guys. Good news for you is Sarah's back next week, so you don't have to put up with me trying to fix up. Oh.